Hi, everyone. My name is Dusan Vuganovic, and I'm a professor of immunology and oncological sciences, microbiology, and the Department of Pediatrics. And I'm also the director of Center for Inborn Errors of Immunity at Mount Sinai in New York.
Hi, I'm Louise Mal. I'm an MD PhD candidate. I did my PhD in immunology in Dusan Buganovic's lab.
So, motivation behind our study was multi-layered. One was that there was an absolute need to understand how does the immune system function in individuals with Down syndrome. Individuals with Down syndrome are an underserved population, understudied population. So we thought that it's pertinent, it's scientifically interesting, and there is a very clear mission of helping individuals with Down syndrome who have multiple immune signs and symptoms that need to be addressing. And that's where really Louise came in.
Yeah, and kind of as Dushan said, because in the MD PhD program, I always want to do research that will has a potential to translate to human health and that can actually help people. And so when Dushan approached me with this project at the beginning of my PhD, I thought it would be perfect because it not only kind of titillated my biological questioning side of things, because it dives into a very interesting part of our antiviral defenses, but it also was looking at people with Down syndrome who, as Dushan said, are an underserved population. And I think that there's a lot of ways in which the treatment for people with Down syndrome can be improved. So I wanted to do my part in that.
I think this is very pertinent because Louise, when she started in the lab, was, as we all were, caught in COVID-19 pandemic. And as she was interested in working and understanding the immune system in Down syndrome, the first thing that she did is she did this survey of thousands, unfortunately, of patients who have COVID. And she essentially found that individuals with Down syndrome have about 10 times higher likelihood of having extremely severe COVID-19. So this was an epidemiological observation that added to a body of literature suggesting that severe viral disease is a problem in Down syndrome individuals. And in part guided by that, really led to this very detailed fundamental immunology observation of why that is the case. So she saw what was going on in the clinic, she saw what was going on in the world, and then came to the lab and ultimately figured out, at least in part, why this is the case.
Okay, so what is the significance of these findings in simple terms? So I think the significance is twofold. First and foremost, I think it tells us something about not only the propensity for autoimmune disease in patients with Down syndrome, but more so tells us something about why people with Down syndrome can be more susceptible to severe viral diseases, which is something that is largely unknown today. And we even have an idea of how this could be targeted therapeutically, because there are drugs on the market that target the pathway that we saw goes awry in people with Down syndrome. And the second part is that it tells us a lot about also just general signaling for this central antiviral pathway that is called type 1 interferon.
Yeah, Louise did such a terrific work here because she disentangled a little bit of a paradox, if you wish, here. Individuals with Down syndrome are inflamed, yet they're susceptible to viral infections. And this study really points to one of the mechanisms through which this is going on. And in essence, too much inflammation causes this immune suppression, and it's a temporal, so it's in time, so it's not always like that. It's like some sort of a sinusoid function, and Louise has so well and so precisely showed that I think this leads to potentially us monitoring health on individuals with Down syndrome and knowing when to intervene with existing drugs, so we can have this less of inflammation but also less of immune suppression, so basically to try and tame these overt responses which go both ways. What are implications? Well, implications are many. First, I hope this is going to be inspiring to others to start studying individuals with Down syndrome at multiple aspects of pathologies that they present with. This has an implication of introducing a different layer in clinical trials with drugs that are now used to tame their immune system. And the clinical output of infectious diseases is not something that's built into these clinical trials. So I think now we can, A, subsegregate individuals with Down syndrome as those who are most likely to benefit from this therapy, both in the context of taming their inflammation, but also helping with the development of severe viral diseases.
Yeah, I honestly don't have that much to add. I think the major implications is um, trying to better understand both the like the high inflammation and the immune suppression in people with Down syndrome, and how actually targeting the same pathway could therapeutically could solve both of these issues to an extent, and trying to study that further.
Well, I
That's a
hope
difficult question.
it is a
Go
hard
ahead,
question.
Jushin.
It is a good question, right? So I think, for me, I think this is an example of how something that people shy away from, and that is, working with a sensitive population, a population that has sometimes intellectual disability issues, population that has caretakers, and including individuals with Down syndrome and other minority groups and other populations that are more difficult, if you wish, to work with and do that in a very rigorous ethical manner, is the right thing to do. That's how we help individuals who are marginalized. That is how we advance medicine. That is how we promote equality in research and equality in medical practice. So I hope this theme comes through as something that others in science and medicine should be perhaps doing more. What should not be taken is that there is no miracle drug, thus, that's going to cure everything. And this is one step forward. We hope extremely important step forward, because if we are going to solve complex issues such as multitude of medical problems that individuals with Down syndrome have, it's one step at a time. And perhaps Louise's studies is three steps at one time, which is why I think it's very impactful. But that's how things work. Things are not simple. Takes a lot of hard work, takes a lot of money to do this. We have to have funding to do this. Um, but both of us, I think, have quite enjoyed it. And perhaps we should thank our cohort of individuals with Down syndrome, without whom, I mean, this would be completely impossible. And they are terrific group to work with, and we've had nothing but phenomenal experiences with that.
I second everything that Dushin said. And I'll also answer this question from a more maybe biological point of view. I think one thing that's a bit difficult and intimidating when tackling a project on Down syndrome, which is caused by trisomy 21, is that it's a lot of genes. It's 200 genes that are triplicated.
Yeah.
So, it's a lot less intimidating to study one gene going awry in one disease. So that makes it a very big challenge in studying this monochromosomal syndrome. So I think what people should take away from it is that studying the impact of each gene on chromosome 21 can be a way to start chipping away at what is going on in Down syndrome. And I think that, careful examination of each gene will bring us a lot. But as Jushin said, what should not be taken away is that this is the end all be all, and that the triplication of IFNAR, which is what we studied, causes all of the immune phenotypes of people with Down syndrome. There's
Right.
interaction between all of these genes on chromosome 21. There's interactions with other genes in the genome. So I think this is a great start, and we have a lot of avenues to explore. But we also want to study all the other genes and their interactions.
Yes, well put. In simple terms, how is this study novel? Well, to date, it was not well understood how type 1 interferon mediated inflammation, so these individuals should be more resistant to viral infections, and indeed they get less frequent viral infections. But at the same time, once infected with viruses, they do so much worse. And we've seen this with COVID-19. This has been reported with a few other viruses. And we just didn't understand why. And now we have one explanation, which we are pretty certain is correct. There's probably multiple other things that are going at the same time because of the complexity of the syndrome. But in simple terms, Louise has discovered why too much interferon can actually lead to insufficient antiviral response. And she even uses drugs that can short circuit this auto amplifying loop in such a way that she rectifies inflammation and rectifies ability to respond to viral disease. So it's pretty remarkable if you ask me.
Thank you. I think that's really the main takeaway, what Dushin said. And the only thing I'll add is that I think this is the first study that looks at the impact of the negative regulation of type 1 interferon on viral susceptibility. And, all of us use interferon to fight viral diseases. So I think this idea that this is a dynamic system that not only turns on, but has very um, intricate mechanisms to turn itself off, and its impact on susceptibility to viral diseases is something that is novel.
Yeah, and perhaps even applicable to general population. It's just that we think, this is a typical response, hence that's the best response. And maybe it isn't. Maybe that can be modulated as well to the advantage of our health, right?
And even as you know, a lot of clinical trials have tried to use interferon, and back in the 80s or 90s thought that it would be this magical drug because it helps cells in a dish fight viral disease, but turns out that it never really worked in humans. So this starts to like answer some of these questions of why interferon is kind
Right.
of like a complex protein to work with and is not the magical drug that people thought it would be. Mm.
You want to
I don't
start?
think I was ever really surprised by what we found because we really, with this study, one thing that I loved about this project is that it was very hypothesis driven. So we had a very strong idea, and then with each step, we kind of tested what we thought was going to happen. And generally, like our knowledge of interferon and its negative feedback kind of turned out to be true with what we expected in Down syndrome. Yeah.
Yeah, I agree with Louise. It was very hypothesis driven. It was background data driven. Um, Perhaps to me, what comes as a surprise is that as Louise mentioned, these clinical trials that are partially effective with recombinant interferon and early phases of disease, maybe this allows us to use the same drugs, in this case, JAK inhibitors to perhaps short circuit the inflammatory response in typical population that can then be better primed to respond to interferon in the context of the antiviral response, right? It's of course even more complicated than that, but the more we understand, the more pieces of the puzzle that we put together, it becomes a better way to perhaps design a clinical trial that's going to have a more positive outcome.
Limitations of the study, I think, are that, as I mentioned, Down syndrome is a very complex disease. And we tried to probe the questions that we wanted to ask. We used mostly cell models. But as we all know, life is much more complicated than cell models. And, people with Down syndrome are at each organ, interferon is going to act a little bit differently. There is going to be viruses that trigger this system. There's going to be other triggers throughout life. And so I think there's still a lot to do to understand how this dynamic interferon response in Down syndrome happens physiologically. We did use some immune cells, but again, in a dish. So I think pushing this to clinical trials, epidemiological studies, and things like that is something that will really round out our findings.
I agree with Louise here 100%. You do everything you can in the laboratory ex vivo, we say that. And then the next step is to run a clinical trial with all this information that we are now empowered with. Right. So the impact of this study, I think, is that of using existing drugs, and we can go into details. What are the options that are blocking interferon using JAK inhibitors? Maybe some sort of a STAT inhibitors, who knows, or TBK1 inhibitors, and designing a clinical trial in which the output is not just taming their inflammation, because we know they have problem with inflammatory diseases, but maybe a secondary outcome should be the incidence of viral disease or severe viral disease, which is what they can suffer from and have much higher likelihood than the typical population. And I think knowing that and knowing the timing of that is perhaps what in a few years can lead to a very specific, tailored treatment that's going to be personalized to the immune state in which that individual is at the time of examination, right? Because just having Down syndrome doesn't mean everybody's the same. Actually, Louise has done another study where we know that's not the case. They can be segregated into a few different groups, and that correlates well with this study as well in terms of that balance between inflammation and immune suppression. So in short terms, hopefully Louise has put us a few steps forward towards a successful clinical trial that will then empower physicians to alter the management of pathology in individuals with Down syndrome.
Let's hope.
Let's hope.
Yeah, well, I guess Dushin is probably better suited to answer this question, as he's leading the lab and I left. But this study really spawned a bunch of different projects, which I'm very excited about.
Yes, I agree. I think this is just the beginning. This is the tip of the iceberg. Louise has implicated, two important genes and implicated how their modification through existing drugs can perhaps help individuals with Down syndrome. But this has also opened avenues into tissue specificity, into understanding how central nervous system functions in Down syndrome in the context of this very inflammation. It has spawned interest into autoimmunity and specific cell subtypes that also Louise has discovered, and we'll talk perhaps next time about that. So there is simply a lot to do. It's, from the scientific point of view, very inspiring and challenging, which is what we like. And I think hopefully it'll inspire also others to take this path of patient-oriented, patient-focused research for betterment of the understanding of fundamental immunological processes, which then leads to better care of the very patients that we work with.